Welcome to my Computational Neuroscience final project. This project consists of 4 steps. The first step will be a review of a couple of papers done by Dr. Avella, which is a former student of the National University. The second step will be the installation of a particular program called Neuron, developed by the University of Yale. The third step will be the download and review of both neuron models. And the fourth step will be the model replication and data analysis using MATLAB. So I hope you enjoy it. To start with the first step, we're going to review the first paper, named External Drive to Inhibitory Cells Induces Alternating Episodes of High and Low Amplitude Oscillations. So what are we saying here? Basically, we know that neurons are connected in networks, and that those networks have also intrinsic properties separate from, well, not separate from, but that emerge on top of neurons' properties. And one of those is amplitude oscillations. They are important because their peaks have shown to propitiate learning and memory activity. However, we don't yet understand where they're coming from. So the paper aims to provide a better understanding of how these amplitude fluctuations and electrical oscillations happen in many brain networks. To achieve it, the authors model interactions between inhibitory and excitatory cells, since these fluctuations are generated by the interaction of those inhibitory and excitatory populations of neurons. What they actually show is that the action potential drive onto inhibitory cells and not excitatory, just inhibitory, desynchronizes the network activity, which is crucial for amplitude fluctuations and hence recognition. So we're going to take a look at that. What the authors did is they modeled the populations of excitatory and inhibitory cells and obtained the amplitude variation similar to the ones found in real data. So what they did is they modeled a network through neuron in a proportion of 80 to 20, 80 excitatory to 20 inhibitory cells, and they found that their results are very similar to real data taken from mice. The results show that the amplitude oscillations might be caused by a temporary decrease in the neuron's firing synchrony. And this is caused by the interference generated by the oscillations of the network and external oscillations that impact the inhibitory population. So here we have an image of a 10-second interval of network activity in the excitatory population, which is responsive to what happens in the inhibitory population. These excitatory cells received an external input of a constant depolarizing current only. They didn't receive anything else. And you can see there the frequency, the firing frequency for that population of neurons. And then we have the same 10-second interval of network activity in the inhibitory population. Now, inhibitory cells are not responsive. They are the source of the fluctuations. So they received as an external input a constant depolarizing current and a train of action potentials. And that is what we have right there. We have the frequency diagram in wavelet, and we've got the frequency graph using cold colors for low frequencies and then warm colors for high frequencies. And we can go to the conclusions, methodology and conclusions. So the model was found to generate a strong fluctuations in oscillation amplitude. So that is high amplitude episodes alternating with low amplitude episodes. The model reproduces high amplitude episodes distributions observed in the prelimbic and infralimbic regions of the rat's prefrontal cortex, which is what we said before. The model actually behaves as real data does. So we have that. And the model reproduces the effect of carbachol, which it was used to provide the, or to induce the oscillations. So carbachol indeed generates high amplitude episodes duration in the hippocampus in mice. And the high amplitude episodes duration was found to have a bell-shaped relationship with carbachol concentrations. That means that the duration of the high amplitude episode increased when carbachol concentration was high. Now, the second paper that we have that we're going to review is called Internetwork Interactions, Impact of Connections Between Oscillatory Neural Networks on Oscillation Frequency and Pattern. So what we have here is basically a paper on frequency variations instead of amplitude variations. This second paper aims to investigate how type and strength of inter-network connections is affected or are affecting actually the target network activity. They aim to understand if the interactions between different networks could contribute to the high variety of oscillation patterns that have been found. Now, neural networks do show a high variety of behaviors. We have already discussed that in terms of amplitude, and now we're going to see that in terms of frequency. But they do have a high range of behaviors that we can see. A network, in terms of frequency, can have a single oscillation frequency or alternate among a set of frequencies, and they can also change depending on the cortical layer we're at. So that is a very rich behavior that we definitely are interested in. The authors created two model networks, just as we did before. The first one with an intrinsic low oscillation frequency, that was 20 hertz, and then the second one with a high oscillation frequency of 32 hertz. Then alternated both networks as the source and target to investigate how network activity was impacted. Of course, in both high and low frequency populations, we have inhibitory and excitatory cells. So we've got a combination of 4 kinds of cells that we're going to see later on in the results. The figure in the next slide shows how network frequencies behave when connected or unconnected for both the slow and fast network. So here we can see that if we have connections, so lower cases, and this is going to be important, lower cases are going to stand for the slow network. So the small I is going to be an intrinsic, it's going to be a low frequency population, and the I is going to stand for inhibitory, and then the E is going to stand for excitatory, and then the capitals are going to be for the same inhibitory and excitatory populations for the high frequency network. And here we're going to see the frequencies at which we're getting results, and then again warm colors are going to stand for the most frequent frequency, if you will, of these networks as a consequence of the connections. Here it is a lot easier to see that, especially in the cases where excitatory cells are connected, the slow and the high speed or high frequency networks are connected, and in the excitatory part of the population, we will at the end see that the first one is actually locking the latter one. So let's see the conclusions so we can have an idea. First off, well, we need to remember that the slow letters stand for the slow population, whereas the capital letters stand for the fast population. And now the blue circles indicate the oscillation frequencies in the fast networks. Their diameters do show the power. Now the red dots are actually the base frequency and first harmonic of the slow network. We can see a couple of those in each graph, and their diameter has nothing to do with power whatsoever. Now for high connection strength between excitatory populations of the slow to the fast network became frequency locked to the rhythm of the slow network. At its base frequency and or the corresponding first harmonic. Now for lower connection strengths, the base frequency of the slow network and a frequency close to the base frequency of the fast network coexisted in the fast network. So as we showed, we found that the slow network imposed its frequency to the fast network when it was selected as a source, especially when its excitatory cells projected to cells of the fast network. Hence, the results suggest that input from other oscillating networks may affect the frequency spectrum of receiving networks, and these interactions may cause a variety of oscillation patterns that we do find in the brain. So this takes care of both papers. We know that we have been discussing the activity of populations of neurons, and now we're going to go ahead and move towards the installation of neuron and the model review and data analysis through MATLAB. Welcome to the second part of the video. As I mentioned before, once we have reviewed the papers, the next step is to download Neuron. So in order to do so, we basically go to the main page of the Neuron project by Yale University. And right there, we can go ahead and download the software. Now, we've got several options here. We have already gone through this process previously, but we've got several options. The first paper was developed in the Neuron 6.0 version, and the second paper was developed in the 7.0 version. And here, where it says other versions, we can always click on prior versions and actually select the one we want to download. In this computer, we had previously downloaded the 6th and the 7th version, but later on we found that it will run, the models will run with no problems when we download the most recent version. So that is the option we actually opted with. We went ahead and selected 64 bits for the particular operating system of this computer for Microsoft Windows, and we went ahead and downloaded the file. When we do, and we actually run the .exe file, we get this folder for each version. So we're going to go with the 7.4 version. And right here, we've got several options. So when we check the instructions provided by the developer of the model, Dr. Avella, we will see that on the instructions he provided, and those are directly in the models, we have an option to compile the modeling files so that we can run them in Neuron. And so what we're going to do is we are going to go ahead and do that for both models. They have also been downloaded here. And in the instructions, we will see that it shows we need to compile them. And how do we do that? Well, we do need to open Neuron to start, the Neuron folder, and then again, run this make Neuron DLL file. When we do, what is going to happen is we're going to be requested to select kind of like a source folder, and that is going to be directly in the model itself. So let's go to the model. We have it right here. We're going to start with this one, the 2012 model. And under the mods folder, we've got several folders. Now we have this folder here, TODO, and there we have moved all the mod folders that we have in the other 3 folders, so that what we want to do is want to make it easier for this to have a path to work with. So now we can go back to Neuron and again run this file, and then we are going to choose a directory we just created for the simulation files. This can be found in users. And we're going to select the actual folder where we have it. And then we make the DLL file that we will later on paste in a different folder. So we're going to give it a couple of minutes for it to finish generating the DLL file. And good, no problems found by the antivirus because it is an executable file. Perfect. And we're done. So now we can actually go to the model. and go back to this folder where we have the compiled files and then the DLL file that we will copy, and then we will paste in the main folder, which is right here. And this will make it easy. It will make it actually possible for us to actually run the simulation, which is right here. So now we can double click on it. Again, the antivirus will analyze it because it's a weird kind of file for it and it doesn't like it very much. But as soon as it's done, it will go ahead and run the simulation for us. So there we go. We've got the windows of the simulation. Let's just put them together a bit. And we can just go ahead and run it for, in this case, 40,000 milliseconds. And here's our control. So we can just go ahead and initialize and run the model. And it will go ahead and run it for a while. Of course, it will take certain amount of time, in this case 40,000 milliseconds, so it can go ahead and finish, which is about 7 to 8 minutes. So while the process completes, and when it does complete, what we will have is we will have a set of data for each of the 100 neurons that were simulated for the first article. Or, well, you know, the proportion was 80 to 20, so it is $100, 100 neurons total. And so we can, once the simulation is done, which is going to take a while, as I mentioned before, or we can run a shorter simulation, but we would get a different result set. What we will get is basically this file, which is the actual data file. I'm sorry, we will get a data file that will be stored here, and with this particular file, we will be able to run the example through MATLAB, which is what we're going to do now. So we double click on it to launch MATLAB. And while it starts, the model will continue to run, and this is what we should have, how it would look like. And then in MATLAB, it's also going to take a while for it to show the results. We're going to see the results later on. As soon as MATLAB launches, it will actually have the file that we, well, the file that we will be working with, the script that we will be running, which contains the paths to the work areas, and of course, well, the code that we will be running. So we're going to show that in a minute. In the meantime, we can always take a look at the simulation that is running. Right now, we're looking at almost 3,000 milliseconds. And right here, we've got the code of the MATLAB file that we're going to run, and we're going to run it through the editor. So this particular process will also take a couple of minutes while the data is actually compiled into a graph, into 3 different graphs that we will see are exactly the same as the ones that were provided in the actual paper. So right now, as it shows, the status is busy, so we're going to give it a minute, and we will be right back with the results. Very well, so after a brief pause, we actually see that MATLAB is done running the simulation. It does show the properties of all the graphs that it is using, and it does show an important amount of warnings that are code related. But at the end, what we actually do get is this figure number 1, which is exactly the same figure that we got for the results of the paper. We have the index, the excitatory cell index. We have the firing frequencies, and the peaks are very clear. And we have the frequency in warm colors right here, exactly in the same way the paper displays them. As to the model, well, the model that we initiated is still actually running, but the results are presumably the same. We are looking at almost 12,000 milliseconds. So the simulation will go on for a while, for a little longer, but the results of the simulation can easily be shown in the MATLAB software. So what we're going to do right now is we're going to do the exact same process for the second paper that is related to frequency and the excitatory cell firing frequency, so that we can go ahead and see those results as well, replicated through MATLAB. So we are going to go through the same process. We can stop this particular simulation, and of course it will show the data that it has. This is for the fast spiking cells population, and this is the pyramidal cells population. And basically what we can do is we will restart neuron for the second paper model, which we are basically going to find at the corresponding paper. So here we have the same mods folder, and we've got all models. We need to make sure that they are all in the same folder. So we're going to copy them into this all models folder, and that is just to make it easier. If they already exist, we're just going to replace them. And we will go ahead and make sure the files are already here in the all models folder, so that we just need to provide one path. Very good. Very good. So again, we go to neuron, but in this case we go to the neuron folder, and we are going to generate SDLL. So we're going to choose the directory that we chose before for users. And we're going to compile them into a DLL file like we did before, so that we can go ahead and run the second model. And as we did before, we exit and go back to the models folder. We have the compiled files and the DLL file that we need to copy into the main folder of the second paper. So we go back here to the main folder and we paste the DLL file. Since it is already there, we're going to replace it. And then we will be able to run the corresponding simulation. The data for this second model will be found in the same run example folder that we will go ahead and open with MATLAB, and that we will go ahead and run in the same way to represent the data through the editor run option. So it will be busy for a couple of minutes. It will display some warnings, and in a couple of minutes we will also see the results, the corresponding results for the second model. Here we go. So here we have the figure for the frequency, the firing frequency for the population, both of the populations of neurons. And we again have the same frequency diagrams with cold and warm colors to represent high and low frequency repetitions for both cases. So basically, this is the way for us to replicate the data of both papers. We have actually been able to study the results through MATLAB and run the simulations through the neuron simulator. So that completes the objective of this particular final project. Again, I hope you have enjoyed it. And well, thank you very much for watching.